Our first speaker is Julian Clarence, Levy Professor of Life Sciences at Columbia University. His research interests center on understanding the regulation of gene expression in yeast and mammalian cells, with a special focus on mRNA splicing and 3' end formation. A fellow of several prestigious academic societies, he is also a senior editor at eLife, and he is here today to talk about his research into the dysregulation of mRNA splicing and how it contributes to human disease. It's my pleasure to start today's talks by welcoming our first speaker, Professor James Manley. Thank you.
Thank you very much. It's a great pleasure to be here at the Crick Institute, which I have not had the opportunity to visit before. And it's also wonderful just to be here in London, maybe the second greatest city in the world after New York. And it's great to be here. So I will start out by just saying a couple words that I think I can speak for my other two speakers. Neither of us know what we're doing. This panel discussion, as it was initially called, was set up sort of with us not really realizing what we were getting into. And you may see that in some of our presentations. We've tried, I think all three of us worked very hard on the airplane coming over here to put something together, and hopefully they'll turn out okay. What I've decided to do is talk about two stories from my lab that are related to how RNA splicing can be dysregulated in different human diseases. And my interest in this topic is sort of what I call from the bottom up. I'm a biochemist and have been my whole career interested in real mechanisms of gene expression. Also with an emphasis on RNA processing reactions, with a belief that these incredibly important processes in cells would ultimately be found to be important in a large number of diseases. And now we know that's true, particularly in the last decade or so with great advances in deep sequencing of genomes of different diseases and RNAs. Lots of old friends of mine, these are biochemical protein factors that we've studied for decades, end up playing roles in important human diseases. And so I'm going to talk today about that, with an emphasis on links to cancer and neurodegenerative disease. So the question is, how is splicing misregulated in disease? And there are several different ways that can happen. The simplest is simple cis-acting mutations in regulatory sequences in genes around splice junctions or in sequences important for splicing. So you would just have a mutation that disrupted a site in the transcript that's needed to carry out splicing, doesn't work anymore, and that leads to splicing dysregulation. There's lots of those. These are the first that were discovered. They're not particularly interesting to someone like me who wants to really understand mechanisms. The more interesting ones are twofold. One, we can change the concentration of splicing regulators in cells, either by increasing their expression, which happens often in cancer, or seeing them sequestered away and having their concentration reduced, for example, in neurodegenerative diseases, as I will talk about. In addition, we can have mutations in splicing factors, and this happens, as I'll start out talking about in cancer, but also neurodegenerative disease. So with respect to cancer, I'm going to concentrate on a kind of what's called often a precancerous condition called myelodysplastic syndromes, or MDS. So what is MDS? It's a disease of hematopoietic cells. We can see here this begins normally in stem cells that go on to a myeloid progenitor and can give rise to neutrophils or erythroid cells following a differentiation process, white and red blood cells. In the disease MDS, there's defects in a step in the myeloid progenitor cell differentiation. There's undue proliferation there often. There's also defects in differentiation. We can have aberrant differentiation and apoptotic cell deaths, which can lead to apoptosis and give rise to cytopenias and loss of anemias and a loss of blood cells. So patients with MDS often die in two ways. One is the disease progresses to leukemia, in particular AML, acute myeloid leukemia, which is very frequently fatal. And if that doesn't happen, the disease course is typically longer, but patients often succumb to, as I said, anemia and cytopenia. So I got interested in this about 6 or 7 years ago when Seiji Ogawa at Kyoto University discovered that mutations in spliceosomal genes are a frequent cause of MDS. And up at the top here, you can see, I won't get into this, a variety of different related diseases, different types of MDS. We initially studied an SR protein splicing factor, serine arginine-rich splicing factors. This was a lot of fun to me because my lab, a good 25 years ago, was involved along with Adrian Krainer's group at Cold Spring Harbor in discovering this family of proteins. And we had worked on SRSF2 20 years ago and determined by a technique called Selective its RNA binding affinity. And what we discovered, along with the lab of Omar Abdelwal at Sloan Kettering, that this defect in the protein altered subtly its RNA binding affinity for different classes of sites in the pre-RNA. So some it would bind more tightly and include that exon, some it would bind more weakly and exclude it. More recently, we've been studying another factor called SF3B1, which is much more of a challenge because it's not an RNA binding protein. It's the most frequently mutated gene in MDS. It's a component, if you remember your splicing, of a complex called the U2 SNRP. It's an auxiliary factor. SF3B is a multi-subunit complex that helps the SNRP bind stably to what's called the branch site. If you all remember that, it's an important element in the first step of the splicing reaction. So SF3B1 is not a sequence-specific RNA binding protein. It is not a regulator like SR proteins. It's a protein required for all splices in the human body, which are hundreds of thousands. Yet in this disease, a very small number, 300 or 400, are misregulated, not something we understand well. Mutations in SF3B1 have been found in a variety of cancers now, all sorts of them, from breast, lung, and others. It's the most frequent cause of a melanoma called uveal melanoma, which is cancer of the eye. It's the most common, as I mentioned, in MDS, particularly in a flavor of MDS called refractive anemia with ring sideroblasts. No time to get into that. All mutations of these and other splicing factors are heterozygous missense mutations. It's because if the homozygous mutation is created, even though it's just a single amino acid change, these are typically lethal for cells. So what does this mutation do? We and other labs have characterized this. What we see here is a switch in a kind of alternative splicing called alternative 3' splicing, which we switch the splicing to an upstream site. So the goal of an outstanding postdoc in my lab has been to understand the mechanism of how the key hotspot mutation, K700E, leads to this dysregulation of splicing. And it was a challenging problem. And I'm happy to say, though, that we're almost at the end of it. And I'll tell you what we know about this. So what we used are cell lines, in leukemic cell line K562, in which another great postdoc who works with myself and Siddhartha Mukherjee, my colleague at the Columbia Medical School, used CRISPR to make the mutation in one allele, and then to that same allele add on two tags, a his tag and a flag tag, that would allow purification of these proteins, which Julian did. Here's a gel. These were subjected to mass spec. And the protein at the very top is something called SUGP1. I'm willing to wager that maybe nobody in the audience has heard of that. Has anybody heard of SUGP1? We have one person. Great. That's more than I would have expected. It's a very obscure splicing factor, and I'll tell you a little bit about it because of what we think is its importance. We had been keyed on this factor previously to this mass spec analysis for reasons I won't get into. And what Julian had done is use siRNAs to knock down a whole bunch of splicing factors that were suggested to us for various reasons. SUGP1 is the one and only one. When you reduce its amounts in cells, it precisely, completely phenocopies the effect of K700E. So we get this switch in alternative splicing by reducing the levels of this protein, which you might expect if it no longer associates well with SF3B1, and that's what we've seen here. So we've characterized this up, did a lot of biochemical experiments. If we overexpress SUGP1, we can drive it back in to the mutant spliceosomes containing this mutation and rescue splicing. So it really seems to be the reason for the defect. So what is SUGP1? Its name is SERP and G-patch domain containing number 1. So what are those domains? A SERP domain is involved in interaction with SF1. This was really nice to us because SF1 actually is involved, and I'll show you the model in just a second, recognizing the branch point sequence, the first step in splicing. In addition, there are some MDS mutations in SF1 as well. They're rare, but that's bringing all these things together. A G-patch domain interacts with dead-back RNA helicases and helps to activate them. So we believe that this interaction is also important for function. And indeed, I won't show any data, but when we mutate the G-patch in SUGP1 and overexpress that protein, it also precisely phenocopies the effect of K700E. So that means there's an interaction with a helicase that when that's disrupted, the splicing goes wrong and we have this disease-related switch. Not much is known about the function. It was found in spliceosome a decade or so ago when spliceosomes were purified and subjected to mass spec. The only published link to any disease is with cholesterol metabolism when there's a deletion of one allele, and that is associated with increased risk of coronary artery disease. Whether this is linked in any ways to myelodysplastic syndromes or cancers, we don't know. To end this part, here's the structure, the relevant structure of SF3B1. It's a big protein, 150 kD. It has what are called heat domains, and these are shown right here in this crystal structure. These are thought to be protein-protein interactions, which is consistent with our discovery that SUGP1 interacts, and the mutation, all the disease mutations are clustered up here. Here's the K700E that we've studied. There's a number of other ones. The most common are in bold type. Julian has made SF3B1 expression vectors with all of those. They're all defective specifically in the interaction with SUGP1 and lead to similar changes in alternative splicing. So we think we've really uncovered the mechanism for dysregulation of splicing that happens with this disease mutation. And here's a model for what we think is going on. Here's the normal case. Normally, there's an interaction with this SUGP1, and this RNA helicase, ignore that number, we don't know what that means, is activated. It's an ATPase, and its ATPase action alters the structure around here, so this SF1 is displaced, and now the U2 SNRP, together with the SF3B complex, recognizes the correct branch site and gives rise to the normal splicing pattern. Either when SUGP1 is mutated in the interaction domain right here, or when it's deleted or its interaction with SF3B1 is weakened by mutation, then this interaction doesn't occur anymore. The SF1 is not displaced. There's no rearrangement that occurs here. And now the splicing machinery is driven to an upstream branch site, giving rise to the change in splicing. So that's a good outline of the mechanism. We need a few more details, but we're quite happy having discovered that out. And it suggests some interesting possibilities, perhaps, in the future for therapy. One question I won't talk about is some beautiful work of Yen Liu, who has figured out how this missplicing can lead to disease. And she identified a misspliced protein kinase in the MAP kinase pathway that when it's disrupted, it causes cells to, the K562 cells, which can differentiate in culture, to aberrantly differentiate and then apoptose. So this is the pathway that happens in humans when they develop anemia, so we think we can identify how that happens. The rest of my time, another 5 minutes or so, maybe 10 at the most, I'll be talking about a totally different disease called ALS, amyotrophic lateral sclerosis. It's a disease maybe many of you here associate with Stephen Hawking, who had an extremely rare disease course and probably disease itself. No one knows exactly what led to his disease, what kind of mutation. He survived a long time. Most frequently and tragically, patients with this disease die within 2 or 3 years of diagnosis. Lou Gehrig on the New York Yankees succumbed to this disease, and in the States it's known as Lou Gehrig's disease. If any of you, and I hate to make any sort of joke about this tragic disease, but if any of you are baseball experts, Kevin Struhl comes to mind, perhaps you can tell me what's wrong with this picture. There's a fault in it, which if people want to know, we can discuss that later. But down below here it says various aspects of the disease. So here is a depiction of the brain and the spinal cord. ALS and another related, at least causally, disease is frontotemporal dementia. It's a dementia that it's not as common as Alzheimer's, but it ranks right behind it. Both of these are on a spectrum in which similar mutations in genes can cause both of them. So here's ALS. This can originate in upper lower neurons or have bulbar onset. I won't talk in the details about that. FTD occurs in cortical neurons. Here's the disease spectrum of a number of genes that are mutated along this way. ALS, SOD1 superoxide dismutase was the first ALS gene discovered. Here are some others I'll speak briefly about. Here's what I'll be concentrating on. C9orf72, right in the middle. Tau, for example, is exclusively associated with FTD. C9orf72 can be associated with both of these. So what is C9orf72? It's now the most common known cause of ALS. And what it is a sort of remarkable hexanucleotide expansion that can be repeated in an intron of this gene in afflicted individuals hundreds or even thousands of times. So we can have in the genomes of these people 5,000, 6,000 or more, 10,000 consecutive base pairs of GC, which is interesting just to think about how that's transcribed and all the problems it can cause by things called transcriptional R loops or formation of G quadruplex structures. Nonetheless, it is transcribed, the protein's made, maybe at reduced levels. There's lots and lots of other genes that are involved at a very low level. Many of these are RNA binding proteins, and this is how my lab got interested in this initially. We also study one of these called FUS, another protein which is an RNA DNA helicase called senataxin that are much less common mutations. But what I'm going to tell you about is work related to C9orf72 and its RNA and its effect on splicing. One important thing we can see here is only 10% of ALS cases can be called familial. That means they're genetically inherited and we are genetically caused and we know the mutation. Sporadic are 90%. And here we can see very few are known. Again, C9orf72 is the most common mutation that pops up in these sporadics. But we can see, if we put all these together, 75% of all ALS cases, we don't know what the cause is. So I'm going to talk now about the work of a former graduate student, Erin Conlon, who wanted to figure out if an RNA binding protein might be titrated by these RNA repeats in a transcript. So if there are made enough of them, can you reduce the amount of the protein that will affect splicing or some other feature in the tau? This had been described previously in myotonic dystrophy as a type of defect. As Erin was doing her work and finishing it up, here was a review that came that was published in 2016, in which a number of laboratories had used really large-scale techniques to purify by RNA affinity all the proteins that would bind to these repeats and characterize them by mass spec, and there's tons of them. We were happy to see a protein called HNRMP-H1, or a very highly related protein, HNRMP-H1, H3 appear in all of these studies because Erin's method was a biochemical one that looked more sensitively at the RNA binding protein. It's a technique called UV cross-linking where extracts are mixed with RNA, and I won't get into the details, but she identified basically just one protein, and it was this one, HNRMPH1. So what are the consequences of binding to these repeats? Here's the properties of HNRMPH1 that have been known from the studies of others for a number of years. It binds to G-rich sequences, which are consistent with the C9 sequence, and it can either act as a splicing silencer or an enhancer, depending on the location of its binding sites. So Erin looked at the literature and found some genes that were known to be dysregulated. And what she found is these were all dysregulated in C9 ALS patients. We saw the same pattern of splicing dysregulation. She then looked at the fate of the protein biochemically. I won't go through this assay. It involves a harsh ionic detergent sarcosyl. But what she characterized was a sarcosyl insoluble fraction. And what we found in this study was HNRMPH, a good fraction of it, so we can see right here, 2 times more than in non-ALS patients, was in this sarcosyl insoluble aggregates. So what happens, and we have more evidence for this, is there's an RNA, C9 RNA protein complex that tends to be insoluble or aggregated. This takes the protein out of solution, reduces its effective levels, and that gives rise to these concentration-dependent changes in splicing. An interesting feature that Erin observed is the extent of splicing dysregulation and insoluble HNRMPH is related to a measurement of disease severity, which is difficult to assess really accurately. What one can look at is the survival time after onset of symptoms would be shorter, and an early age of onset, or evidence of more serious or stronger disease. This correlated with greater insolubility and splicing defects, which suggests doesn't prove a causal role for sequestration in disease. So this sort of summarizes what I've told you so far. I'm now going to finish off in just a few minutes talking about the other 75%, or the 90% of cases in which there's no mutation, there's no C9 expansion in these unknown red cases here, which are the bulk of ALS. So what Erin wanted to investigate is, might there be a related defect here, even though we don't know the genetic cause of the disease? So together with our colleague Neil Schneider at the medical school, who's the head of the ALS Center at Columbia, we obtained postmortem samples from 50 sporadic patients, and Erin characterized them in a number of ways. Here's looking at splicing of HNRMPH dependent exons, and what you can see here, these like C9s behave pretty much like the real C9. They have splicing dysregulation. But another half of these patients were just like wild type. They didn't show that splicing dysregulation. So we could bind all these sporadics, which are most of the cases of ALS, into 2 groups, like C9 or like control. Now, Erin also used the sarcosyl assay and found that HNRMPH was insoluble in the like C9s, as was expected, but she found something else that was unexpected, which was that lots of other proteins also showed this same insolubility. These include some of these. If some of you are familiar with ALS, you'll recognize these proteins as also being associated with the disease. SMN is associated with another neurodegenerative disease, which has been linked a little bit to ALS. These are all insoluble. Here's some controls. The C9orf72 protein itself and a histone are not in the insoluble fraction. We then got together with scientists at the New York Genome Center to do RNA-seq and splicing analyses of a large cohort of patients. And what we found here is there was massive dysregulation in the ALS high insolubility group that mirrored C9. When ALS patients had high solubility, when the protein wasn't, they were like controls. It was a remarkable total lack of any splicing differences. Interesting, frontal temporal dementia patients show the greatest splicing dysregulation amongst all of them. More recently, we've looked at these splicing dysregulations more carefully with Don Rio at the UC Berkeley and his postdoc, Qingqing Wang, who's developed a new annotation-free software for analyzing splicing. And what we've discovered is the most frequently disrupted event is called intron retention. So splicing just fails. And that's consistent with the insolubility of lots of different splicing factors. As with C9s, again an analogy, and I'm just about done, the like C9 patients that don't have a C9 mutation are much more likely to have a risk of developing FTD. So this is, again, another important link. So to conclude, here's the last model slide that we think now of ALS perhaps as a multi-RBP proteinopathy. We can see right here a continuum in which free proteins often exist solubly. This little flag might constitute a disordered region in these proteins. Physiological granules can occur, SNRPs, nuclear speckles, other things, these are natural. However, various perturbations such as the C9 expansion and other mutations, stress and injury to the brain have also been thought to lead to neurodegenerative disease, can cause an irreversible aggregate. And that's what we're talking about, where these proteins are titrated out and cannot perform their function. Here we have a depiction of that from diffuse RBPs, perhaps reflecting phase transition coupled with a toxicity boundary that leads to these aggregates right here. And this shows up in different disease progression or different disease susceptibility, ALS at one end, and FTD and perhaps other motor neuron diseases at the other. So thank you all for listening. The work I talked about, the cancer work, MDS, Jian Zhang did all the biochemistry, Yen Liu has created cell lines and done great work on her own. I collaborated with Sid Mukherjee at the medical school. Azra Raza is the head of the MDS Center at Columbia. Abdullah Ali helped with this. Raul Agaban and Zalki Li did bioinformatic analysis. As I mentioned, Erin Conlon, a former graduate student, did most of, not all the work I talked about on ALS. She worked with a great undergraduate, Julius Davis Parada, and a postdoc who helped with the work. Neil Schneider is the head of the ALS Center, as I mentioned. These are our colleagues at the New York Genome Center who did all the RNA-seq analysis. I didn't have time to talk about another RNA binding protein that we think binds to the repeats, which is a component of the transcriptional remodeling protein, PRC2. We're studying that with Tom Check. Don Rio and Qingqing Wang helped with our most recent splicing analysis. Thank you very much.
Questions, Jim?
So I just wonder about the first story and SREBP1, which is super interesting. So do you have more insight into the specificity for that small subset of exons now? The multiple branch points probably a part of it, but is that all of the story?
That's a really good question that we and others have been trying to think about and looking for characteristics in the sequences of those introns. Nothing really shows up if that's at all convincing. So we really don't know. Most of these alternative 3' splice sites are what are called cryptic splice sites. That is, they don't seem to really be used naturally. So we're not really having a switch in alternative splicing. Somehow with this mutation and the effect on SREBP1, you're using a sequence that's not typically utilized. Usually they're like 20 nucleotides upstream of the natural one, almost always. There's some exceptions. We're studying one cryptic site that's 83 nucleotides downstream, which when you try to think about it mechanistically, that's tough. So your question's a really important one, and we don't know the answer yet.
Sorry, following up the same question actually. Do you see any overlap with the deletion of the splicing factor with the effect of spliceostatin?
Not completely. Spliceostatin, for those of you who don't know, we haven't done those experiments. Others have, is an inhibitor of SF3B1, so it's more like a loss of function. That's not really exactly what's happening here with SF3B1. So I say we haven't done that, but I think the overlap's not great with the effect of that inhibitor.
Sorry, can I pass it to you?
I just realised that a lot of people can't hear the questions. Sorry.
Yeah, fascinating how you have a mechanistic explanation for the disease phenotype. You alluded briefly to that you'd have an idea how to use that, turn this around to treat the disease. It's not obvious how you can do that. Can you say anything more about that?
About how this might be used to treat the disease? The idea, this is Julian Zhang, the great research associate's idea, if we can map precisely by structural methods where SUB-P1 and SF3B1 are interacting and see the difference with the mutation, if we could somehow design a peptide that might come in and prevent the mutant form, design a mutant peptide that might block the interaction with the mutant form of SF3B1. This is his idea how they could possibly do that. And we're working with a colleague at Columbia, structural biologist, to try to get these structures right now.
Time for one last question for Jim? Anyone got a question?
So Jim, in the second part of the work, the group of patients that didn't form into this splice alter, do you think they're a group that are maybe represented by the SOD mutations and oxidative stress?
That's a great question. That's definitely one possibility, maybe the simplest one, that we could maybe divide all ALS patients into like SOD1 or like C9, because SOD1 does not show these splicing defects. The other possibility is maybe there's a gradient of these splicing changes that occurs with time or something, or maybe we just miss the right part of the brain, and maybe there are these changes that we missed in those. I sort of like what you suggested, that there's these two classes with like SOD1 perhaps in some of them.
Thank you. Very much. Just join me in giving James another round of applause. So yeah. Our second speaker is also from Columbia University, where she is the DaCosta Professor of Biological Sciences. She's known for her work in the characterization of the transcriptional activator and key tumor suppressor protein P53. She's here today to give a talk on the regulation of the P53 network in cancer cells. So please join me in welcoming our second speaker to the stage, Professor Carol Prives.
Can I use this too?
Use the pointer with the clicker won't hold. Right. Let's get that down back on.
Okay, but I should open it up now.
Yeah.
Okay. Okay. And then put it onto slideshow, is that right? Okay, thank you very much for the invitation. Like Jim, and very likely also like Kevin, I had no idea what sort of presentation to give you. Like Jim, I'm going to also ask the audience if they've ever heard of this protein. So I get some of you have, and perhaps if you haven't, then Karen Vosten, who's an absolute spectacular leader in this field, should be really unhappy, but I'm sure you have heard of it. And so actually when I put this talk together, I really didn't know. It was supposed to be about gene expression, and I'm very interested in gene expression, and P53 is actually considered a model eukaryotic transcription factor, and I found myself being very pedantic in preparing this talk, so I'm going to be very much like the college professor-teacher type for the first part of this short presentation. Then I'll tell you some recent, but all published data, including some very recently published data. So this is supposed to advance the slide, and it doesn't. Does this? Yes. Okay, but this works. Okay, so we know that P53 is a major tumor suppressor. I won't go through it very slowly. We know that it is because it's frequently mutated in human cancer. A very important component of understanding the tumor suppressive function of human cancer, P53 in human cancer, is the fact that there are very unfortunate families with very high predisposition of cancer, of a certain set of cancers at very young ages that have germline mutations in P53. Mouse models are incredibly important. If you knock P53 out of a mouse, that mouse will get cancers, again, of a certain type. And there is some in vitro data that's showing that P53 can suppress the ability of oncogenes to transform cells, and P53 was discovered on DNA tumor viruses, and these tumor viruses, we know, have evolved to abrogate P53 function. Okay, I keep forgetting that this isn't working. What's interesting about P53 and its mutation in human cancer is that it seems to be quite unique as a tumor suppressor, in that rather losing the protein, very frequently you get missense mutant forms of P53 protein that are present at very high levels in cells. And we know now from many people, including Karen's work, that these missense mutations are not neutral in cells. They actually have oncogenic properties. And I'll get to that a little bit more at the very end of this talk. We also know that the P53 protein is extensively modified. It's modified by phosphorylation, by acetylation, by netylation, and other modifications, glycosylation, etc. And we believe that all of these modifications do have functional consequences for the protein. We can show that in biochemical assays. It's a little more difficult to prove it when you use mouse models, partly because of the complexity and partly because when we study mouse models, we very often don't do very careful analysis of all the tissues of the mice, and therefore we don't know the roles P53 is playing in every single tissue in our body. But we do know that P53 is we know a lot about how P53 works in cells, and this is the result of work of many different people, including our own group. And we know that it's a sequence-specific transcriptional activator. And we know that there are myriad genes that are regulated by P53. If you toggle the levels of P53 in the cell, or you compare P53 plus and minus cells, you will see a big difference in genes. Actually, about, I'd say, a couple of thousand can be shown to be affected by this P53 status in cells. And at least half of these actually are downregulated. And yes, there's scant evidence that P53 is a transcriptional repressor. We believe it's an activator, and at least many of us believe that a lot of the repressed genes are so repressed because P53 activates either a protein that itself is a transcriptional repressor, or a microRNA that can downregulate gene expression, or a long non-coding RNA that can regulate gene expression. So I'm of the sort of old school that P53 is really a bona fide transcriptional activator. And there's a myriad genes that have been discovered that mediate P53's many activities as a major tumor suppressor. I should say right now that we don't really know how P53 works as a tumor suppressor. Every time we think we've got the answer, somebody does some experiment to say, no, it's not this, it must be something else. So I'm cleaving to the idea that it depends very much on the context and the way you do the experiment and many different roles P53 has in cells. The fact that it is heavily mutated, however, is consistent with the idea that there are multiple really interesting genes that are affecting many different aspects of cell biology, from cell death and cell cycle. Among the first types of cellular responses shown to be mediated by P53, senescence DNA repair, this new trendy topic, ferroptosis, which we're working on a little bit, metabolism, very important. Karen's work has been incredibly important in that area, stem cells, microenvironment, and so on. So and what's interesting is that you never have just one gene that's mediating an outcome. There's multiple genes. And the reason why some of these lists are short are probably because there are genes yet to be discovered that are mediating its activity. Certainly we know that there are more genes in ferroptosis that are functioning as P53 intermediaries in this kind of cell death. So I thought, here's where I'm getting very pedantic. How do we know a protein is a P53 target gene? These are what I believe you would have to show, that it in fact the gene has a binding consensus sequence, that purified protein can bind to it, but not a mutated tumor-derived protein can bind to it, and that if you mutate the sequence, you don't get binding. You should be able to confirm that in vivo using chromatin immunoprecipitation. and that you should have good controls for these experiments. You should also show that this binding is associated with a fairly rapid response of RNA that should probably be done by looking at elongating RNA, and that you should see chromatin-associated changes, chromatin marks that are important that would be associated with this gene promoter. And you should also ideally be able to use some kind of gene editing to actually either knock P53 out of the cells or ideally and more challengingly mutate the P53 binding site and show you no longer get a response. But we also know, we also know that when P53 is activated, depending again on the context, some genes are activated and some are not. So the selective responses of P53, I think, are very important in trying to understand how this protein works. And here's where I somewhat self-servingly gave a list of possible ways in which P53 might select its target genes. And I say self-servingly because we happen to have published papers over the years that have addressed these. And let me tell you, this is a very small number of the important papers that have been that have been reported that show that P53 can be selective in the genes that it activates. The levels of the protein themselves can determine whether a cell is going to die or arrest. High levels of the protein are more likely to induce cell death than low levels of the protein. P53 cooperates with many different factors. We discovered Cas-CSA1 many years ago. P53 has 2, possibly 3 transcriptional activation domains, and they function to differentially regulate many genes. TAD1 is the most important one. We've discovered a protein that's actually part of the nucleoporin complex that regulates messenger RNA stability that cooperates with P53. A lot of work has been done on showing modification of P53 can differentially affect its interactions with members of either DNA or the transcriptional machinery. And very recently, we've been working on the oligomeric state of P53 and how that itself might also be selective. So one of the problems we deal with in studying this protein is not only how it makes choices, but how different cellular stressors, which I didn't get into, actually help it make those choices. And so here's an experiment that was done by 2 members of the laboratory, Kaushik Reghanath and Vitaly Fomin, who basically just took 2 isogenic cell lines that are very extensively used in the P53 field. These are colon carcinoma cells that are wild type for P53, but from which P53 was deleted many years ago, pre-CRISPR. Now it's possible to knock P53 out with considerable ease. In fact, we've remade these cells. These are P53, therefore essentially null cells. And if you compare the response of the plus versus the minus cells to various different kinds of stressors, Nutlin being the most, I say, pure one in the sense that it simply separates P53 from MDM2 and activates the protein. These other proteins, doxorubicin, neocarcinostatin, and cisplatin, have their own set of impacts on cells. And what you do is, in each case, P53 levels rise, and a lot of different genes are affected. This is an RNA-seq analysis, and you can see that each one of these stressors has very unique responses that are P53 dependent. Shares some responses with other stressors. A small number, I'd say about 15% of these we'd say, core responders that will respond to any one of these stressors. They're the same. And so you can see that this is very complicated. And I think teasing out, these are all chemotherapeutic agents, at least doxorubicin and cisplatin are. And so understanding the P53 response in the context, and this is just one type of cell line, in the context of different tissues is going to be, I think, the big challenge going ahead for people studying this protein. So finally, as if that weren't enough, we know that P53 can do other things as well. P53 has a role in the cytoplasm as well as in the nucleus. It's been shown to be associated with either with proteins that affect mitochondrially based apoptosis. It's been shown to directly interact with several of these proteins. And I was skeptical when I first learned about this several years ago, but I'm a complete convert now. I think P53 has multiple roles, and there are many examples of other proteins like that. that can do things in the nucleus, but they can also do significant things in the cytoplasm as well. So now I'm going to move over for the second part of the talk, and here I show some work from our laboratory, which is addresses this idea I presented at the beginning, which is that P53 is not only a tumor suppressor, it's also actually an oncogene. So when it becomes mutated, it's not only do you lose the wild type function, but in certain cases with certain P53 mutations, you actually have a protein that has oncogenic activities in cells. As I just mentioned, Karen did some beautiful work on this a few years ago, showing that it affects Rab recycling. I hope I've got it right. We subsequently also found an interesting role for P53 in how it might, as when mutated, affect oncogenesis. And this is just, again, some sort of pedantic slides. Why do tumors select for mutant P53? Loss of the wild type protein, the dominant negative activity towards the wild type P53, and then gain of function effects. What is the idea of gain of function is that mutation of P53 is not equivalent to simply losing it. These actually acquire oncogenic properties, and here's some evidence for it. First of all, unlike many tumor suppressors, these mutants are present at very high levels in tumors, and they've been shown to have novel transactivation targets and protein-protein interactions. If you overexpress mutant P53 and P53 null cells, this is an experiment that was done a couple of decades ago by Levy, you can actually get more aggressive and more frequent tumor formation than simply P53 null cells. And whereas wild-type null P53 are extremely tumor-prone, these tumors rarely metastasize. If you knock in a common missense mutation of P53, you will get different forms of tumors, not at a very high rate, and these tumors can often metastasize. And in fact, the presence of mutant P53 in some human tumors correlates with more aggressive and more metastatic tumors. So, and finally, I'm not going to go through this very detailed slides, but there's a lot of evidence, and this is actually a very old review, but a very good one, I should say, just listing some of what was known about how these different mutant P53s might actually function in cells. So this is the work of Will Prives. He was an MD PhD student in the lab several years ago, and I'm not going to go into it in great detail other than he was working with breast cancer cells where you could inducibly reduce the single mutant P53 in those cells. What I mean by single mutant is that there was no wild type P53 in the cells. When that mutant P53 was downregulated, a high proportion of these cells, almost 50%, went from a very malignant morphology to a more normal-looking acinar, hollow lumen-like morphology. And this led Will, and collaborating with other groups in Arnie Levine's lab, to actually do a gene expression analysis. And surprisingly to us, the pathway that was most significantly affected when you change the levels of mutant P53 was the what's known as the methylonic acid pathway, the pathway by which cells synthesize cholesterol, also there are adducts that cause glycosylation, coenzyme Q that plays a role in the electron transport chain, and adducts that are necessary, known as protein prenylation, for anchoring RAS and RAS family members to the membrane. So there have been several inhibitors based on these very important activities, and perhaps the most famous are the statins that prevent hypercholesterolemia. And in fact, what we showed that when you treat breast cancer cells with statins in a 3D protocol, you greatly limit their growth and their malignant morphology. And so this pathway, in fact, has been playing, when it's deregulated, it's been shown to play many roles in cancer, and many of the products of this pathway have been shown to be linked to cancer as well. And I think I went through that to some extent. This is a wonderful review from Linda Penn. If you're interested in this, she really covers all the bases, she and her colleagues. So this brings us back to gene expression. Will's hypothesis was that this whole pathway is coordinately regulated by a single transcription factor. And that transcription factor is called SREBP2. And it basically, almost all of these genes contain binding sites for the mature form of SREBP2 in their promoter. And Will's hypothesis is that mutant p53 is somehow working with SREBP2 to increase expression of these genes. And his data support that hypothesis. He was able to show that mutant p53 was present at the binding site for SREBP2, called the sterile response element, at several of these genes. This is your chromatin immunoprecipitation. This is a sort of a low resolution promoter scan of HMG-CoA reductase with a peak right at the SRE element over here. He also showed that if you ablate SREBP2 with siRNA, you got less mutant p53 binding. But what he didn't show, and what we know, is that the way mutant p53 interacts with this promoter is different. So this is, again, the same scan of the same promoter, and this is SREBP2 itself. And you can see that it very sharply peaks right at this element over here. I'm sorry, it's missing here, but this is the SRE element here. Mutant p53 is very broadly distributed. So we think it's a different kind of interaction, essentially. That yes, it may be recruited partially by SREBP2, but it's doing something different at this promoter. Which leads me to the next topic, which is the work of a subsequent MD PhD student in the lab named Neil Pfister, who was very interested in how mutant p53 mechanistically might regulate expression of its genes. And he didn't want to work on the cholesterol biosynthesis pathway, so he screened the list of genes that we showed that mutant p53 could upregulate. And he found, oh, I pressed the wrong person's, that was actually Kevin's talk. You better be careful when you look at your talk. I didn't mess it up. He screened and he found that VEGFR2, itself a very important gene in cancer, it's a receptor for VEGF, which is very important in stimulating angiogenesis, which in turn is very important in maintaining tumors. He showed that VEGFR2 indeed is a mutant p53 target. This is messenger RNA with high versus low levels of mutant p53, and this is the protein over here, and he did a lot of other experiments supporting this. VEGFR2 plays a very important role in many aspects of cancer. It plays a role in maintaining survival, migration, proliferation. It is a very good target, potential target for mutant p53. And what Neil did, and I'm not going to get into great detail, was to do a very careful analysis of the binding of mutant p53 to the VEGFR promoter. This is the proximal region, right, adjacent to the start site of transcription. And he did an analysis of essentially nucleosomes present at that very region. And he showed that when mutant p53 is high, there's lower levels of nucleosomes right in this region. And he was able to basically show that mutant p53, therefore, is required for displacement of nucleosomes at the proximal region of the VEGFR2 promoter. He then discovered that mutant p53 can associate with several members of a chromatin remodeling complex called SY-SNF. And this is just a little sort of summary slide for that. SY-SNF chromatin remodeling complex consists of a number of subunits, including these core subunits, BRG or BRM. I should say, although I'm not going to talk about it here, we're studying the relationship of this chromatin remodeling complex to components of the cholesterol biosynthesis pathway, hoping to bring together these two subjects we've been working on. And basically, what Neil was able to show was that immunoprecipitating mutant p53, he could co-immunoprecipitate several of these subunits, and developed a hypothesis as well as some evidence showing that mutant p53 and BRG/BRM work together to stimulate chromatin, essentially nucleosome eviction from the VEGFR2 gene. He then did a sort of global analysis, collaborating with Vitaly Fomin in the lab, and showed that a large number of genes that are regulated by mutant p53 also require the SY-SNF chromatin remodeling complex. Not all of them, but a large number of them. So this was the idea, was that you would get, you would increase, one of the functions of mutant p53 is perhaps to work in some way we don't fully understand, to stimulate not so much the recruitment, but somehow the activity of this chromatin remodeling complex. and we're hoping to study that more. It kind of explains the difference in distribution of mutant p53 at the promoter. In other words, it's more broadly associated with many promoters, probably because it has a secondary set of interactions with this and possibly other chromatin remodeling complexes. So the final little bit of data that I'm going to talk about is a very recent, is basically an offshoot, and it turned out to be, I think, an interesting offshoot of our discovery that mutant p53 can stimulate expression of the genes in the methylonic acid pathway by which cells synthesize cholesterol. And one of the things that's very interesting about the reciprocal relationship between wild type and mutant p53 is they really seem to be, in fact, reciprocal. Genes that are often actively stimulated by mutant p53 tend to be repressed by wild type p53, and conversely, many genes that have been shown to be wild type p53 targets are actually very often down in cells that are overexpressing mutant p53. So it seemed pretty obvious to Sung-Won Moon in the lab, whose work spearheaded this work, that perhaps these a repression function of wild type p53 might be to downregulate the cholesterol biosynthesis pathway. And in fact, this is the sum of this work. This work has been very recently published. And it was, again, a collaboration with many different people, including, very importantly, Scott Lowe and two very outstanding members of his lab, Chun-Ho Huang, who's now left his lab, and Shauna Houlihan, who's done some very beautiful work recently, as well as collaborations with others at Columbia and with the metabolomics core at the University of Colorado. And I should say, Angelo knows Karen. Is that right? Is he not maybe a colleague of yours from a long time ago? Yeah, because he pointed this out when we were chatting. At any rate, Sung-Won was the spearheaded leader of this group, and his work is very beautiful or very impressive in my belief. And I now have to tell you something about SREBP2, as well as the other SREBP, which is SREBP1. Both of SREBP1 regulates many aspects of lipid metabolism. SREBP2 is more or less dedicated to cholesterol biosynthesis. This is the work of Brown and Goldstein, Nobel Prize winners who studied this pathway, studied how it's regulated, and their work has led to important health-improving drugs, spectacular biology. SREBs are held in the ER as inactive large precursor proteins, and then when the cell is deprived of cholesterol, they are transported to the Golgi. They are subjected to two separate cleavages, which then releases the mature active form of the SREBP2, which gets into the nucleus and binds to the sterile response element that I alluded to before. And so I don't show you here, but what Sung-Won showed was that in any cell with wild type p53, there was less of the mature form than there was in cells that lacked p53. And these were very controlled cell lines that were isogenic. They either had wild type p53 or p53 had been deleted from them. It wasn't just general correlation. And he had many other lines of evidence to show that wild type p53 somehow affects maturation and inhibits maturation of this process. And the challenge we had to figure out was how this worked. We tried looking at any one of these genes and we found nothing. Changing their levels, p53 did not affect any of these different proteins that are here. So for a number of reasons, Sung-Won read the literature very carefully and discovered this protein, ABCA1. This is actually best known as an exporter of cholesterol from the cells. So it would be very unlikely to be a candidate, except there was one paper published by a group in Japan a couple of years ago who had found that ABCA1 actually can also retrograde transport cholesterol to the ER. And the ER is very, very sensitive to levels of cholesterol. Changing the levels of cholesterol in the cell by as little as 5% can activate this pathway. And so what Sung-Won hypothesized that maybe ABCA1 is a p53 target gene, and indeed it is. If you increase the levels of p53, you increase the levels of ABCA1 message, and this is also ABCA1 protein. You also can find p53 associated with a likely p53 response element at the ABCA1 promoter. And if you use a transcriptionally inert version of p53, this is a transcriptional mutation of the p53 activation domain, you no longer can increase the levels of ABCA1 or p53's famous target p21. So then the idea was to see whether we could in fact confirm the observation that ABCA1 itself is regulating SREBP2 maturation. And this is an important experiment, I think, that Sungwon did, because he took p53 knockout cells and he activated ABCA1 using an agonist of RXR, because it's regulated as well by these receptors. And he discovered when you actually induce ABCA1, you get a significant drop in the levels of the mature SREBP2. So he was able to confirm the idea that ABCA1 is actually playing a role in blocking maturation of this protein, and ABCA1 is a p53 target. This work, which was all done in cells, was confirmed by some, I think, really nice mouse models that were brought in Scott Lowe's lab, where Shun Hao and Shauna were able to show that if you induce tumors, and these are specific liver tumors, hepatocellular carcinomas, using an overexpressed MYC and loss of p53, they were able to show that this led to overexpression of the methylonic pathway. And furthermore, this overexpression of the methylonic acid pathway genes were actually creating a dependency of these cells. Because if you either inhibited these genes from being expressed or used a statin, you no longer got tumors. And this was not the case if you set up another tumor suppressor model. It was at least specific to p53 under that condition. Furthermore, if you looked very early, before you actually see tumors form, you can actually do single-cell RNA-seq, you saw very rapid response. In other words, loss of p53 was correlated with very high levels of these genes, even before you saw tumors. So it's a very initial stage in tumor formation. And finally, we were able to show that loss of ABCA1 itself is tumor suppressive. And in fact, that correlates with increase of mature SREBP2 in these cells. So this just basically summarizes what we believe. We believe there's an important connection between p53 and this methylonic acid pathway. Mutant p53, as I've showed, you can somehow stimulate it. We don't fully understand how it can. At the same time, wild-type p53 represses it, we believe, at least one way through activation of ABCA1. And in either case, deregulation of this pathway is correlated with tumor formation. And that's it. This is my not-so-recent picture of our lab. And I always like to show it in context of how international we are. And I can see here that this is probably a very international group as well. And that's one of the beautiful things of science. Thank you.
Thank you very much, Charles.
Some questions for Carol, please?
Thank you, Carol. That was lovely. Right at the very beginning, it was really interesting to see how many genes are regulated by treatment of cells with nuclein, but not any of the other things. Are you surprised by that?
No. Why? Because I think just raising the levels of p53 as it does without causing any p53 modifications is enough to allow it to occupy some promoters.
Yeah, no, I would imagine that those would also be activated by all the other activating signals.
Yes, you're asking what question, why is nuclein unique? And that's a really, right? That's what you're asking.
It suggests that the signals that are sent by the other stresses is counteracting that.
Yes, it's as if that were the case. And I don't know the answer to that question, but it's a really interesting observation, isn't it? Yeah, I don't know. I mean, many of these genes do a lot of things without p53. And nuclein does a few things. I mean, it's not completely p53 dependent. I don't know if you found this or not.
in?
So, third speaker, third and final speaker of today, is Professor Kevin Struhl, who is the David Wesley Geiser Professor at Harvard Medical School. He is also a senior editor for Eli. Whilst the presentation is being set up, his research uses a combination of techniques to address fundamental questions in the regulation of transcription and chromatin structure in yeast. He is also interested in elucidating the transcriptional circuits behind cellular transformation and the formation of cancer stem cells. And it looks like his presentation is ready, so I'll hand over and welcome Kevin.
So this is the microphone?
That microphone there.
Okay.
Yeah. Thank you so much.
Well, thank you very much for the invitation to speak here. I have an unusual connection to Francis Crick. He was my landlord when I was a postdoc in England at Sixcroft Lodge. And probably those of you who have ever followed my work will know that I'm known primarily for gene regulation, transcriptional regulation, chromatin, yeast molecular biology. I'm not going to actually talk about that today, although if you want to ask me questions later, feel free to do so. I'm going to talk about something different. I guess I've really gone down in the world. I'm now working in the part of our lab is working in the RNA field, and that's what I'm going to tell you about. So the first part of this talk is, this is all done by two amazing people in my lab, Joe Geisberg and Zalmar Mokhtadir. They're longtime members of my lab. And you'll see from the work, there's a lot of creative experimental new techniques involved in this lab and very complicated informatics that is not standard, which they've done all of it. Even though none of them were trained as informatics people, the informatics they're doing is quite sophisticated. So the first part of the talk relates to genome scale identification of mRNA stabilizing and destabilizing elements and the role of the poly A tail in secondary structure mRNA stability. So we got into this totally accidentally. We were, in course of our transcriptional experiments, we're using a technique to remove RNA polymerase from the nucleus. That's a technique in yeast called anchor away, where you throw in a small molecule, and the RNA polymerase, or any protein that you've tagged, is sucked out of the nucleus and trapped in the cytoplasm. So you can study the function of any nuclear protein. And we were doing this for RNA polymerase II for a lot of different reasons. And we realized, well, we could actually measure mRNA half-lives doing this because it's a very good way of removing the polymerase. And we coupled that for reasons that were kind of random at the time. Instead of just looking at individual genes, we actually looked at 3' isoforms. So we took the RNA that we got after the depletion of the polymerase, and we used a DT primed based method, sort of a 3' race type method, to map RNAs. So we were actually measuring the half-lives of 3' isoforms, not just the gene, a single gene itself. So the first main result we found is that, I should also say that in a slightly earlier work, we showed that the average yeast gene has about 60 different isoforms. And so here's a pattern of a typical yeast gene. These are all the different isoforms from position 1 to 400 from the stop codon. You see there's a lot of different ones. There's about 60 typically. Some are used more than others. But in this experiment, what we could see is that there are occasionally times where adjacent isoforms can have extremely different half-lives. So if you look here, for example, at isoforms A and B, at time 0, they're equal. But 2 hours after you've done the depletion, you see that B is still pretty stable, is pretty around, and A is much less. And if you compare that to A, B, and C, it's the same thing. So this says that isoforms that are 1 or 2 base pairs apart, these are virtually identical because they're a single base pair or 2, can have very different half-lives. And we have hundreds of examples of this. Sometimes the upstream one is more stable, sometimes the downstream one is more stable. And something I'll get into is, you see the relevance of this, 3' terminal U residues, the extra residues at the end, if they're 3' terminal U residues, that can help stabilize isoform half-lives. That is, in the cases where they're more stable, that is an important component. Now, that's an interesting curiosity, but the more powerful use of this is shown here, and that is to basically look at this in the following way. So we basically discovered something we call an isoform cluster. And the isoform cluster is a series of isoforms that are all very close together, and they all have the same half-life. So all these isoforms have this half-life, this cluster is this half-life, and this cluster is that half-life. Now, it turns out that what's important about that is that that allows us to map stabilizing and destabilizing elements, because if you compare these two clusters, the difference is this bit here, and you see you go from a 20-minute half-life to a 9-minute half-life. So that's a destabilizing element. And conversely, if you go from here to here, this extra region here in green tends to go from a 9-minute half-life to a 35-minute half-life. That's a stabilizing element. So we can actually map them completely over the entire yeast transcriptome this way. Now, before this work, there had been very, very few elements like this mapped, because they were mapped in a kind of clumsy way, individual ways with chimeric proteins. This is a general way to map them over the entire transcriptome. Now, for those of you who are not geneticists, this is a very appealing genetic kind of experiment, because this looks like classical deletion analysis, right? The difference between the two things. But unlike almost any other deletion analysis, this is done with natural isoforms. We're not making mutants here. These are the natural isoforms. So there's no issue about creating new constructs or whatever. This is actually the real guys. Okay. So overall, we identified about 560 stabilizing and 800 or so destabilizing elements. So if you look at the sequences here of these kind of elements, the destabilizing elements, we don't really see much of anything. But in the stabilizing elements, some proportion of them turn out to have a homopolymer U sequence. All right? Now these, again, are not things we added. This is in the natural genes. And many of the genes here have a poly U sequence that is mapping to the stabilizing element. And to prove that this really is the stabilizing element, what we did was an experiment where we took several genes, actually 6 different genes here, and we in each gene we have 2 isoform clusters that are equal half-lives. And what we did was we replaced the intervening region of that neutral sequence with a poly U element that was from one of our guys. And what you can see here is that if you look at this gene, for example, here, the lower things are, in cases with the black, we have basically the upstream cluster. And of course, the upstream cluster is not affected by putting the U's in there, so that's the same. But the red guys are the downstream cluster. And what you can see is that if you have the poly U sequence in place of the normal sequence, it's a much slower half-life. And that's true for 5 out of the 6 genes that we tested. So what this says is that the poly U sequence is sufficient to confer extra half-life. And I won't go into, because this is supposed to be a short talk, I won't go into all the gory detail, but so it turns out that what is the poly U sequence likely to be doing? Well, the most obvious possibility is that it's hybridizing to the poly A tail. And what I've shown you here are the most extreme examples. The poly U element is the most extreme example, but we've actually gone through the entire data set and looked at a hybridization of the poly A, of the sequence we see to the poly A tail. It is a very striking relationship, which is that the more you're able to hybridize to the poly A tail, the more you're stabilizing the half-lives. And it turns out that what the destabilizing elements are is, if you think about all the possible intervening sequences, there's an average sequence that will allow you to hybridize to the poly A tail. The destabilizing elements are mainly sequences that are just less good at hybridizing to the poly A tail than average. So they get mapped as destabilizing elements because you're comparing it to the overall stability. It's not that there's some special thing destabilizing, it's just, so we think that the poly A tail has just a completely new role for the poly A tail, that it controls to a large extent mRNA stability by hybridizing. That's the idea. Now, I'm again not going to, in the interest of time, I'm not going to show you this, but we also did experiments where instead of putting in the poly U tail region, poly U region in here, we also put in a GC-rich hairpin. And the GC-rich hairpin also stabilizes. So it isn't only the poly A tail that matters. Other forms of secondary structure will also stabilize the mRNA. Now, what's the cause of this? Well, one obvious possibility relates to the protein that binds poly A tails, namely the poly A binding protein. And it turned out David Tollerley had done a genome scale experiment where he looked at various proteins binding ability, RNA binding proteins, genome-wide. And what's shown here is, because we know the genes that have the poly-U regions, we basically compared all genes or genes that had a poly-U element. And we looked at, in the upstream and downstream isoform regions, we looked at how much binding there was for poly-A binding protein. And so what you can see here is if you take all genes, whether you're upstream or downstream, the poly-A binding protein level is pretty much the same. But if you look at the poly-U related genes exclusively, you see that there's much greater downstream binding than, there's much greater upstream binding than downstream binding. So you have less poly-A binding protein when the poly-U element is there, presumably because the poly-U element is hybridizing the poly-A tail, therefore blocking the poly-A binding protein. And interestingly, these are two different degrading enzymes. One is from the SREBP and one is from the exosome, and you can see the same exact pattern. But as a control, this is another exosome component, but this is a nuclear exosome, not the cytoplasmic where the thing is, and there we don't see any difference. So genome-wide mRNA stabilizing elements are associated with low poly-A binding protein and other degradation factors. And that's a genome scale experiment. This is in our own case. I told you about our alleles that we made, that we put the poly-U elements in between, artificially, between natural sequences, and we see the same results. If you look here, if you look at the wild-type genes, you see upstream and downstream equal poly-A binding protein. This is an RNA chip type experiment. Whereas here, with the poly-U elements, you can see for these genes where we inserted the poly-U element, you can see that there's, in fact, lower binding to the downstream guy. Okay, so the obvious model is that if you have less stable, you have poly-A binding protein binding here, and the factors, other factors are there. If you hybridize to the tail, you have less poly-A binding protein and less of these factors, and that gives you more stability. Now, the last thing I'll say in this half of the talk is that one of the nice things about this method we can do, this RNA, the anchor away method, is we can do it under any growth condition we want. And so what's shown here is the beginnings of a whole new project, which is we actually analyzed our mRNA stability exactly the way I told you, but under different conditions. And what's shown here, for example, is if you look at these two different conditions and compare the ratio of the half-lives of these conditions, if you take the gene that's in blue, you can see that the half-life is the same no matter which condition you use. But if you look at red, you see this region right here, this element right here, is much different in half-life under one condition than the other. Okay, so this is a condition-specific stability element. It's clearly making you much more stable, but only under the right growth condition. Here's another example with a different growth condition, and here's yet another example for a different growth condition. So we have many hundreds of these, and we're trying to work our way through how this is actually involved. So I'm going to switch gears a little bit. I've told you a lot about the poly-A tail. I've talked about a lot of structure, but I haven't shown you any structural anything yet. Okay, so we decided we really needed to study the RNA structures of these isoforms, not just all this genetic stuff, even though I like genetic stuff better. So we invented a new technique. We call it DREADS, for determining the structure of individual 3' isoforms in vivo on a transcriptome scale. And it's using DMS footprinting. Okay, so DMS footprinting has been used for 1 million years. We didn't invent that part of it. DMS modifies single-stranded A and C residues, and if a non-reactive A and C residue is non-reactive either because it's double-stranded or because a protein is bound to it. Okay? And an important fact is that a DMS-modified residue blocks reverse transcriptase. So if you treat cells with DMS, which other people have done before us, you get the modifications. But what we've done that's different is we're doing paired-end sequencing on a 3' based method. So if you imagine a sequencing read that we get, here's our sequencing read. This end tells you which isoform it is to the nucleotide, and this end tells you where the modification was, because it's blocked from extending. And so what you do is you take all these paired isoforms and you do it. And so if you take all the data for a given isoform, you will get a complete modification pattern of that isoform. So what you're essentially doing is you're getting structural information from individual isoforms. Okay? So that's pretty useful for a lot of reasons, but the result that was really dramatic is that structures of closely related isoforms can differ dramatically over 300 nucleotides. So here's an example of a particular gene, and this is the biological replica. We're looking at isoform plus 145. Okay, you can see in red and blue the replicates are virtually identical, correlation over 0.9. But now, using the same samples, it's the same data, it's the same exact samples, we're comparing the isoform at plus 145 to plus 150. So these are two isoforms that are only 5 bases different. Look how different this is. You see lots of reds with no blues, blues with no reds. The correlation is 0.41. And in fact, we have cases that are even a lot worse than that. So that says that closely related isoforms can have dramatic structural differences. Now to show you this in an overall summary, what we've done here is we're comparing replicates that are in light gray, and what we're doing for all the replicates, these are now probably 20,000 examples of things here, we're looking at what the pairwise correlation is between the data, like I showed you before. And you can see most of them have very high correlations. Most of them are over 90%, a lot of them are in the 0.8 to 0.9 range. That's the pattern you see. But now if we look at paired isoforms, these are the ones that are of interest, you can see that there's a class of them, maybe 20, 30% of them, that are extremely poorly correlated. I mean, this is really, really bad correlation. And it turns out that the poorly correlated isoforms, of course, get higher and higher as the distance gets longer and longer. But even at distances like 3 and 6 base pairs, a significant number of those guys, and we're talking hundreds of examples here, it's not one or two things, have really bad correlation even when they're only a few bases apart. So many paired isoforms, including 10% of closely spaced ones, are structurally different. Now, an important aspect of this is that, remember, these isoforms only have a subtle difference, but the structural differences are going over the entire 3' UTR. That's a pretty striking result. Now, what I'm not going to show you, I don't think, is, some reviewer hassled us to death about this. So we did an experiment where we actually engineered a mutation in the region between two isoforms. And if we do that, we get exactly the same result. That we can artificially put a mutation somewhere, and we get the same thing that in many cases, it doesn't always happen, but in some cases, you see a dramatic difference over the whole structure. Okay, now, I told you that when you see a structural difference, it can be due either to duplex formation, or it could be due to proteins. Now, naively, you would think, how could you possibly do these, you do structural folding programs, for example, and you have isoforms that differ by 2 bases, they're going to give you the same results. So how do we get this? So the experiment we did was to compare the in vivo data I just showed you with the exact same experiment, except that what we did was we purified the RNA first from the cells, the exact same cells, purified the RNA, and now refolded the RNAs in vitro and did exactly the same experiment. And what you can see here is that, as I showed you, in orange we have, around 10% of these pretty closely spaced isoforms showing very different. This number here is the percent of isoforms that are less than 0.3 correlated. So these are really different structures. And you can see that I told you before, we have about 10% or so of these closely related isoforms are different. But if you look in vitro, there's almost never. Okay? So that says that, especially for these guys, almost all of those structural differences are due to RNA binding proteins, not the intrinsic RNA-RNA interactions. And if you look at distances further and further away, you can see that as the distances get further away, then of course you also get structural differences in RNA interactions in vitro. But you can see that in every case, you always see more structural difference in vivo. So what this strongly suggests is that many of the structural differences that you see in vivo are due to the RNA binding proteins there, not so much the actual intrinsic structure. Although, of course, the intrinsic structure does matter too, especially when you get out here. But for the close ones, it's almost all RNA binding proteins. So how does that work? Well, what's the obvious? Things are very similar. So the most obvious case is that at least one important component of that would be the poly A binding protein, because that's binding right near the region that's different. So we invented another technique. We call it clip reads. This is a classic precipitation with poly A binding protein. So we pull down the RNA, but instead of just doing what people usually do to it, we then do the same kind of 3' based thing. So what we're essentially doing is we're measuring the ability of poly A binding protein to bind to different isoforms. We're essentially doing binding experiments on all the different isoforms. Now you would have naively thought, at least initially, we certainly would have originally thought, but well, they all have poly A tails. What's the big deal? It's going to bind all of them. Well, it turns out it doesn't bind all of them equally. So I didn't show you is that it can actually vary over 100-fold, depending on which isoforms. Most of them range in about a 5 to 10-fold range, but there are significant differences in the ability of an individual isoform to bind the poly A binding protein. So what's shown here is we're comparing two classes of situations. We're comparing classes where we have differential poly A binding protein, differential binding between two isoforms, and that's greater than 2-fold, or similar binding, which is less than 1.2-fold. Two classes of neighboring isoforms. They either have different poly A binding abilities or they have similar. And if you then ask how does that relate to isoforms with structural differences, you can see that there's a dramatic effect of structural differences correlating with differential poly A binding protein. But interestingly and importantly, that only occurs on isoforms that are closely spaced. If the isoforms are far apart, then you don't see any effect of that. So differential poly A binding is associated with extensive structural differences for closely spaced neighboring isoforms. All right. Now, what else do we know about the structure? What we've done here is, based on the structural information, we wound up essentially uncovering a 2-part structural motif. And basically, the structural motif is that this region here, between -24 and -18, is unstructured, and this region from +8 to +16 is structured. And if you put those components together, you get a motif score. What you can see is that the closer you are to this motif, the longer the half-life. So this 2-part structural motif, which is actually based on structural information, real actual EMS information, is linked directly to more longer half-lives. And interesting, this region here between -4 and -18 is evolutionarily conserved for structure. So that suggests the functional importance of RNA binding proteins that facilitate degradation. Okay? Because it's the unstructured stuff that correlates with long half-life. This is correlated with structure, and that means that suggests that because it's evolutionarily conserved, that there will be RNA binding proteins in this region that facilitate mRNA degradation. Now, the last thing I want to say about this, in fact, the last thing I pretty much say in the talk, is that does this actually make any difference in biology, all this stuff I told you about? And it's very hard. We don't have any easy way to say, well, here's a really clear example. So what we do instead is to use evolutionary considerations to decide what is functionally meaningful or not. And so what's done on this slide here is we've taken all these different situations, and we look at the region that is different between the isoforms, and we ask, what is the conservation score based on that's conservation scores of a whole bunch of yeasts? What is the conservation of the red region here? And so we can, again, compare structurally similar isoform pairs and structurally dissimilar isoform pairs. Again, two broad categories that are very different. And what you can see here is that structurally similar pairs, there is a bias towards being not conserved versus conserved. Whereas it's the opposite for structurally dissimilar isoform pairs. The ones that are very structurally different is the opposite. And there's a very significant p-value. So what that says is that the sequences between the distinct isoform, structurally distinct isoform pairs, are evolutionally conserved considerably more than chance. So that suggests that in a general sense, these things mean something. Okay, we can't, at the moment, we don't have a specific example to say this is doing that. But we think in general, we can say that this overall idea has biological meaning. So the last slide I will show you is how we think this is working. This is the sort of speculative model. We call it a domino model for dramatically different structures. So in vitro, if we compare isoform A versus isoform B, which differ by these two N residues there, there's no difference. But if we compare it in vivo, what we think is going on, what's our idea, is that we know that in some cases you have a differential PAB1 binding, right? So let's say this one is a lot stronger than this one. Well, if this protein is binding more strongly and having an effect over here, that's going to affect the ability of these nucleotides right nearby to either engage in RNA-RNA interactions or RNA-protein interactions. And essentially, once you've changed it over here, you now have something different that gets read out by different proteins. Okay, so once you've differed it over here, now, for example, I've shown you secondary structures arbitrarily. You can see that the secondary structures here are different because some of the nucleotides over here are no longer available to that. And once you start changing that, then that'll affect which protein can bind, and once you change which protein can bind, you can change that. So the idea is it's essentially a domino effect, is that once you have one single difference, and we think this would be kind of the initiating difference because it's got to be something pretty close to the difference, we think once you initiate a difference like with PAB1, that you keep, by virtue of the fact that you've now taken some nucleotides out of play, you affect both the structure and the protein. And as you build it up, you get more and more. And that's why we think that even though the differences are very subtly over here, we think it extends out over the whole 3' UTR because all the proteins are involved in 3' UTR and they're all being affected. Now the last thing I want to mention is an evolutionary thing, which is that during evolution, we now know that subtle mutations can have a big effect on all these different isoforms. Doesn't always have it, but it can have it pretty frequently. And obviously during evolution, subtle mutations are going to happen in 3' UTR. And what we think is that in a lot of cases, that will dramatically change the structures and the constellation of associated proteins that are there. So that what looks like a simple mutation that doesn't make a lot of difference in the 3' UTR, in some reasonable number of cases, can have actually quite dramatic effects. So that we think is one of the other interesting things we found from it. So that's sort of the end of the talk. Again, I want to point out that this work was all completely done by Zarmek and Joe. Really quite impressive work. I didn't really show you a lot of the informatics that went into doing all this stuff. It's really non-trivial. And they've done all of it, from conceptually doing it to running all the programs. And none of these people were trained in informatics. So it's actually quite a nice piece of work. So I will take questions.
Question at the back there? Hi. So you talk about poly-U binding to poly-A and therefore displacing PAB binding PAB. But does that displacement actually prohibit the cells, the message from being circularized and translated? Therefore, there's a inverse correlation between stability and protein translatability. And therefore, there might be some isoform that fold back are less translated and sent to, say, P-bodies.
Okay, so we have not looked at translation here, so we don't know. I mean, there's no question this can affect translation. Another thing we do know is, I didn't talk about it, we developed another method that essentially is able to map the length of the poly-A tail in an isoform-specific fashion. There's quite a bit of variability, more than you would think. There's condition-specific effects on that. So that we do know, but we haven't really linked it up yet. We have not really looked at translation. It's not totally obvious how to do that. So we have not looked at isoform-specific translation at all. Yes, Bert.
So, Kevin, one of the characteristics of the SCI2 mutation is that you can actually translate poly-A free messenger RNAs in that mutant.
What mutant is this?
SKI2.
Oh, yeah.
Okay, so this is an RNA helicase,
yeah.
and an obvious consequence of what you've been telling us here is that these RNA helicases are disrupting either the protein RNA interactions or the secondary structures of RNA in the ends. I was wondering whether this technique...
I don't think we really know that. I mean, because we've not looked at this. We actually are doing this.
So this is my question, is whether mutations in PAP1, which is difficult because it's essential, or SCI2, which is not, affects these structures.
Yeah, so we haven't... There's a lot of things on our list.
Right.
We haven't... That's one of them. We haven't just not... We might have done some of them so far. We have not really totally analyzed that. I mean, there's only 2 people doing this, and so there's like... We're doing it under different conditions. They've invented a lot of techniques. So that's a pretty important experiment. And we actually are also trying to do it with the the PAB1 mutants, as you say, they're essential, but there are mutants that are that are still viable. So that's a definitely important experiment. We haven't done it yet.
And this differentiality you saw between isoforms, is that restricted to the area at the 3' end, or does it actually extend further into the gene? I saw the regions went to -100. I suppose that's compared to the poly-A cleavage site, so that goes sort of into the coding region, right?
Right, so I didn't say this, but at least in yeast, and at least because we see very little polyadenylation in the coding regions, like next to none. So we can't really look.
No, but that's not what I mean. I mean the differential sort of structures you see. These differences are all restricted to the 3' end, or can you not map it further up?
No, you can map. In fact, it's not explicitly restricted. It's restricted mostly by the size of the fragments you can get. So some of the structural information actually is in the coding region, but most of it isn't. That's limited by, yeah.
Sorry, were we looking?
By comparing these different isoforms, you sort of found new mechanisms how you can stabilize or destabilize the transcript. And my question is, does that affect sort of global stability of all the transcript of a particular gene? Because there are some genes that the transcripts are always more stable or almost less stable. So would a very unstable transcript, for example, just simply have no possibility for hybridization for loop formation?
So we didn't really look at that way. I mean, obviously there are a number of things that go into mRNA stability, and so it's not just. I mean, we never would claim this is the only
obviously,
thing
but it could be one thing that also.
So there are obviously genes that forgetting about the isoform-specific differences, there are certain genes, even if you compare isoforms, that definitely have lower half-life than others. And I don't think we really looked at that way. The advantage of doing this, the trouble with doing that, is that you have so many variables, you have no idea what to look for. And the advantage here is we really could link it very directly to, that's really the power of comparing the individual isoforms. It's really taking advantage of classical genetic approaches, even though there's no genetics here, because we're comparing 2 things that are extremely similar and trying to see what the differences are. That's very useful. That's a very old-fashioned Jacob-Monod style of doing experiments. If you just take the overall pattern, it's really difficult to figure out what's what, because there's just too many things. So we didn't really even look. And it would not even be obvious how to even do that. How would we, for example, if we wanted the poly-A tail to be important, how would we even average it out over the whole thing? It's very difficult to do that. So we didn't even try. I'll admit, we didn't even think of it.
Okay, so there's one last question at the back, and then I think we'll just continue these discussions over the drinks outside. So.
Hello. What I was wondering is where you have these 3' isoforms that are very, very close together, but have really different half-lives. Are these genes, do they contain introns? Are they spliced? And could these be splicing errors that are destined to be degraded?
Okay, so this is yeast, Saccharomyces cerevisiae, that has very little splicing. So the vast majority of genes in this organism are not spliced at all. There are genes that are spliced, but not, and some of the ones that I'm talking about are. But I don't see how this would really relate to splicing, because remember, all these things are being captured by virtue of having a poly-A tail. So even if you didn't splice properly, I'm not sure that we would see that one way or another. I mean, because we're not sequencing the region. We're not, even for the genes that are spliced, we're not looking at that region of the RNA, so we have no idea what's going on. I should also say that not only do most yeast genes are not spliced, but the ones that are spliced almost always have one intron. There are very few examples of multiple introns in a yeast. There are examples, it's not 0, but it's, I think, less than 10.
Okay, so thank you everyone. Can you please join me in showing our appreciation for our 3 speakers?